Hello once again, and a very warm welcome from us here in Singapore. My name is Clarice Yeo, and I am a second-year medical student at the NUS Medical School of Medicine. We welcome you back to COVID-19 Updates from Singapore. This is the second in a series of 12 webinars presented by NUS Medical School of Medicine, National University Health System, and Global Outbreak Alert and Response Network. The COVID-19 Updates from Singapore weekly webinar series will provide viewpoints and insights from a panel of leading experts in infectious diseases and related specialties and disciplines. It is my pleasure to welcome our program director, who will also be the moderator of today's update. Recruited to establish an infectious disease training program in Singapore, he was the first infectious diseases head of department in the Communicable Disease Center in 1992. He is also a visiting senior fellow of the Courage Fund at the university. Ladies and gentlemen, Dr. David Allen.
Thank you, Clarice. Welcome again to the second of our 12-part webinar, COVID-19 Updates from Singapore. I hope this broadcast finds you and your loved ones safe and well. The episodes have a characteristic pattern to them. We will begin with an update in regional and international epidemiology of COVID from Professor Dale Fisher, followed by our expert guest this evening, Professor Wang Linfa, who will present his lecture. Then we'll have a question and answer period with panelists including Professor Wang, myself, and Professor Fisher. This will then be followed by updates of key aspects presented this evening by and an update of current events by Professor Fisher. and then we'll have a preview of next week's episode guest expert. We welcome you to send your questions and your comments of how we can better serve you. It's my pleasure now to introduce Dale Fisher, who's Professor of Medicine at National University of Singapore, Yang Lulin School of Medicine, Senior Consultant, Division of Infectious Disease in UH, and Chair of the Global Outbreak Alert and Response Network hosted by World Health Organization. Dale, over to you.
Thanks, David. Hope everyone can see the slides okay. And hi, everyone, again. There's a great number, and I'm sure many friends and colleagues out there. Just let me indulge you for the first 5 or 10 minutes to give a background update of what's happening in the world. As you know, this is the Johns Hopkins University well-known source of epi information. And what I'm doing here is comparing last week, or 8 days ago actually, 1.4 million people affected, 2 million now. So the exponential rise that we understand. Some people get shocked that it's now going so fast, but this is what we understand in epidemiology. The death rate's gone from similarly from 83,000 to 137,000 on this side. You can see now the top countries affected. US gone from 400,000 to over 600,000. Spain, Italy, Germany, somewhat leveling off because they were affected early and naturally shut down. China with about 500 new cases. UK kicking along with a later shutdown from 55 to 99,000. A later shutdown, and B, a recent ramping up of tests. I wanted to just drill down. If you take the same website and you just click on the United States, it's just a nice reminder that this is not outbreaks in New York and Washington. This is a whole sort of mesh of outbreaks. This is not one outbreak. This is worth just reflecting on a little bit, that every city, every town, every state governor, every Lord Mayor, every provincial leader, they're all going through what we're all going through. And I think it's sometimes nice to reflect on the people that aren't in the media, that are equally dealing with their own problems and stresses. WHO data that I receive regularly, the China numbers that absolutely dwarfed by the European numbers, and the yellow ones being the American numbers. What they've done most recently in their figures is added this black line, which is the deaths, and the axis is down here. So you can look now and see that there's about 70,000 cases a day and 6,000 deaths. So that's where we are there, and this is by mostly by Africa, by WHO region. So again, the African numbers still not climbing, partly as surely under ascertainment, but yes, they are the last continent to be really affected. But they'll have a big problem with testing. They'll have different looking outbreaks. Here's the Americas. Here's China. This interesting one here. This is the cruise ship, so they don't really fit into any region. But you can see how almost all the cases were diagnosed before there were deaths, and you can see this long tail of deaths out to sort of 2 months later, and that's a message for I guess the intensivists in the audience that if and when you have a shutdown, it's going to take a long time for your health system to recover, because to clear those beds one way or the other does take some time. Otherwise known as, we're very good at keeping people alive for a long time. This last one, the Western Pacific region, I mentioned last week, this little double hump here, this first hump being the Korean outbreak in Daegu. The black line doesn't really move during that, and that's because we know the death rate during that particular outbreak within Korea was very young people. So this is another website that I recommend. This Our World in Data. You can spend a lot of time browsing around here, but I just use this one because we all like to talk about flat curves. Of course, you have the flattest curve if you're shut down. So you've got China. This one's South Korea. They haven't shut down. They've just according to my colleagues there, it's just a community acceptance of what's needed. So I guess it's a sort of a shutdown, but not by stealth, if you like. Singapore and Japan's numbers are climbing. I said last week I was very worried about Japan. They didn't seem to have a lot of the measures in place. And I was also worried about Singapore because I knew what was happening in the dormitories. We'll talk more about that a bit later. Australia basically shut down. I hear they've extended for another month. Unlocking is going to be really hard. That strategy is yet to be confirmed, and China are leading the way, and they're going to we're going to learn a lot from them. Down here is Taiwan and Vietnam, which have got very good public health measures in place and very flat curves, albeit with at least Vietnam with a certain degree of social restriction. So just flicking to Singapore, just to focus on really this number. You can have a bit of a scan everywhere. You can see how all our various hospitals are involved, with NCID naturally taking the lion's share. But this is what's most important. So today, 15th of April, or yesterday, was 447 cases. 38 were community, 5 were work permits but not living in dormitories, and 404 were dormitory residents. And this will feature a little bit in my discussion at the end of what's happening in the dormitories. So we've got two epicurves going on here. This first one is the imported cases, and they're basically none for several days now. And I guess that's what's expected because transmissions in other countries have fallen as well as that. Here's our community numbers. So in total, it's 900, but it's over 2,000 in the dorms. And even though it really only started, at the end of last month, they've skyrocketed. These are very densely populated dormitories of foreign workers who do most of the manual labour in Singapore. There's 43 of them in all, with the largest being around 24,000 people. So you can just see how they're all affected, the sheer numbers. And with that, David, I'll hand back to you. We'll talk more about the dormitories at the end. Thanks.
Great, Dale. And we apologize for the technical difficulties. You're not able to previously see my lovely face. I do want to remind our participants, there we are, our audience, to send your questions through the question and answers section at the bottom of the toolbar. We appreciate those, and we will use those part of our question and answer period that follows our guest lecture. It's now my privilege to introduce Prof Wang Linfa. He's a professor and director of Program in Emerging Infectious Diseases, Duke-NUS Medical School, Singapore. Title of his talk is COVID-19 from Bats, Pangolin to Coronavirus and Serology. Linfa, over to you.
Thank you, David. Yeah, so thank you, Dale and David, and thank the COVID-19 Updates Forum, and also welcome all our audience. Yeah, so I thought that, it's a hard topic to cover, so I just put the keywords there. What I'm going to cover is really from bats, pangolin to the coronavirus in general, and then I end up to just really say now, at this critical stage of the outbreak, serology is going to be playing a more and more important role. So this is the outline, a few dot points there. I may start with just tell you, audience, that what I have been doing in responding to COVID-19. And then I discuss, is this really sort of a surprise to people like myself? And then the question of, is this SARS or not SARS? When it first emerged in China, that was the hot topic. And then as a virologist, I would do some talk about the virus itself. How much do we know, right? And then the topic about origin. Is it bat virus? What's the intermediate host? and the law of serology, as I already said. We end up with the lessons. I have been involved in emerging zoonotic virus, especially from bats, for the last 25 years. So I have made penned a comment in Lancet, and I will summarize this. So I start with my personal experience. January 14th to 18th, I was actually in Wuhan on ground zero. And I said it's by accident, means that I did not go there to invest COVID-19 because at that time it's not called COVID-19. And I have strong collaborations with the Wuhan Institute of Virology. So I'll go there every year for 3-4 times. So I was there to discuss bat coronavirus, but not COVID-19 specifically. So when I came back, the Singapore government has not started quarantine, but as a scientist in this field, I thought, my God, I was in Wuhan on the ground, and I have a full-packed airplane direct flight from Wuhan to Singapore. So I self-quarantined myself, and that gave me a lot of opportunity really to work on committees. WHO and OIE invite me, and so I was on that famous WHO international regulations, health regulations emergency committee. That really made a decision: do we call it emergency of public health international, concern, and international significance. Sorry, and then before we had the first case, the Singapore government has an inter-ministry task force, and the scientists and clinicians also had this national response team. So I was on the team, and I was assigned to be leading the originated serology. Interestingly, that was one day before we had the first case. So we were ready even before we had the first case. And then, very different from my past experiences, I have been conducting like a close to 100 media interviews. Last but not least, I was quarantined, and the MOH was keep calling me. So I say, 14 days later, can I go back? And say, you can go back, but we're going to monitor you for 17 days, And then I decided to quarantine myself for 17 days just to play safe. Because of that, I had a lot of time in those days, to read and to really digest. And I think, to be able to read both English and the Chinese media really helped me to understand and go into this. So now the topic of, is that a surprise? Are we ready? For me, it's not a surprise. So I came to Singapore to lead the emerging infectious disease program in Duke-NUS. And then 2013, after MERS, I got interviewed by the Straits Times. So this is an interview, and if you enlarge the quote in the newspaper, said you can bet on it. So this is my direct quote. I'm almost certain that in the next 10 years, a new killer virus spread by bats will emerge. As a scientist, when you get your prediction right, you should be happy. But in this case, I'm kind of sad, because I predict the outbreak, but I did not predict the scale of the outbreak. I thought nothing can beat SARS. SARS was bad, and never predicted that we can get something worse than SARS. Not only in newspapers, right? So I have this grant, is a collaboration between Singapore and China. So the title is Combating the Next SARS, almost like emerging infectious disease outbreak, by improving active surveillance, mainly in bats. And last year, I was asked to write a review in current opinion in virology, and the conclusion is to say, among the known unknowns, I always use the term of almost like fighting terrorists. You have the known knowns, known unknowns, and the unknown unknowns. So by now, COVID-19 is known unknowns because SARS was the unknown unknown. So I say, among the known unknowns, I really think the bat-borne coronavirus will be the top of the list for us to watch out. So all this to demonstrate, for scientists like myself, work in the field, we have been predicting this, so it's not a surprise to us. So is this a SARS or not SARS? Now, that depends. If you go to SARS, it's a severe acute respiratory syndrome. For COVID-19, now we know majority of patients, 85% has been quoted as mild disease. And now in this stage of the outbreak, we're even talking about asymptomatic infections, right? So if you judge by that, then COVID-19 is not SARS. But as a virologist, then you go to the virus. The virus is 80% identical genetically to the SARS coronavirus. And in virus taxonomy, we have family, genus, and species. So before COVID-19, the ICTV already have officially designated this SARS-related coronavirus, or SARS-CoV, as a species name. So COVID-19 definitely belongs to the same species. So, I use the analogy, so we're talking about if you're in humans, we're in one species, right? If whether you're Chinese or Indian, you're just different ethnic groups. So SARS, and COVID-19, it's the same virus. And the naming virus is very, very complicated. So for every virus, looks like we could not get it right. So initially it was 2019 nCoV, and now COVID-19 is the disease, and SARS-CoV-2 is SARS-CoV-2 is the virus name. So now a little bit of virology, right? I mean, you have seen this, images all over the world now. Everybody knows how coronavirus look like, right? So the name came from this spike protein on the surface. If you do a section, it looks like a crown, like this. And the genome, so this is the genome. And the coronavirus is unique. I would say that, it's a large genome of RNA, single-strand RNA. And it's not drawn to proportion, but really, these are the amino acid numbers, 7,000, out of 10,000. So 70% of the genome is really coding for this big protein machinery responsible for replication and transcription. In the structure proteins, then you have 4 of them, the S for spike, E for envelope, matrix, and the nucleic capsid. So these are the 4. So these are 4 essential. If you delete any of these, the virus will not be able to survive. Then for SARS and for COVID-19, we have these ORFs. ORF stand for open reading frame. Just means that we really don't know what their function. So we give them a number, ORF3, ORF6, ORF7, ORF8. So ORF8 is the important one for the context of the SARS and the COVID-19, then I'll come back later. Okay, so if you enlarge, the spike protein is the most important because that's the protein really make the first contact with the cell host. So if you enlarge that, then the green region is the receptor binding protein. So in the 3D structure, as you can see, that's the green region. This is very important because they make a direct contact with the receptor, which is the in human, this is angiotensin converting enzyme 2 or ACE2, and that's really important. So if you look for virus mutations, the first is you look into IBD if there's any major mutations. Okay, so in terms of virology, as I said, coronaviruses is really unique. It has the largest non-segment RNA genome, around 30 kilobases. But flu virus, we have segmented, so you have different RNA genomes. It goes from, less than 1 kb maybe to 3 kb. And other RNA virus like dengue, you can go to, close to 7 to 10 kb. But no other virus has that large genome, and the coronavirus is unique. And we think that may also be, a reason that bats can handle large RNA genome virus, and that's why bats are such a good reservoir for coronavirus. You see this statement, the SARS-CoV-2 is genetically more stable than SARS-CoV and also MERS-CoV. And it's in the WHO report and also in many other scientific publications. We know now, as I said, it is a SARS virus in the same species. The other important really criteria says if it's the same species is to see whether the virus can use the same receptor. And as I said already, COVID-19 uses the same receptor. And the most close related virus so far is a bat virus, and at the genome level, it's 96% identical. That's kind of impressive, When we first discovered SARS in bats was 92% identical. It took us 10 years to find another virus which is 99% identical. But the story sort of evolves. Initially, we say it's a bat virus, and then many reports says actually pangolin carries a COVID-19 virus in the same species of the SARS-related coronavirus. But importantly, the receptor binding domain of the pangolin virus is even closer to the COVID-19 than that virus. And I'll come back to this later. So this is the first paper really published in Nature, have a very deep characterization of the virus and the receptor. So anecdotally, I like to point out, the first 2 authors are former postdocs trained in my lab, Duke-NUS in Singapore. So now they are all in Wuhan Institute of Virology. So, and then if you can see from these, phylogeny trees, as you can see this nCoV at that time called, the bat virus, this is the bat virus RATG13, is the only one really close related. But really to illustrate this is important, if you look, the SARS virus is on top, the Beijing 01 is here, and the MERS virus is down here, okay? So I always say that if MERS and SARS can spill over and jump to human and kill, and now COVID-19 can, and look at all these backbone virus in the tree, right? So that's what I say. Again, if we don't change our way of trading and farming of wildlife animals, and we don't change the culture, then I think COVID-19 will not be the last outbreak of backbone coronavirus. More will come. In terms of the evolution, there are many papers. I'm just going through three. So in terms of the virus, so the first one is done by informatician mainly in China. And the paper is, I have to say, very, very technical and very dry. So I'm not going to bore the audience tonight. But the conclusions are important. Basically, this is the first paper to say that the first known cases is actually December 1st rather than later December. And importantly, it had no link to that famous Huanan seafood wholesale market. And then the major sort of cluster, the second batch really on December 10th, and they had 3 cases and 2 had no market link. Then the major cluster we learned from news, early January is these 41 cases in Wuhan. And that really sort of rang the alarm bell to say there are severe pneumonia of unknown etiology, and discovered to say that a novel coronavirus could be the etiology. So, and also that because majority of the cases, 28 were linked to that market. That's why everybody believed, just like SARS, start in the market. What this paper tried to say is that, yes, there is a large cluster, majority is linked to the market, but even in that 41, 13 had no link. So the market now, the theory is, may not be the original source of human transmission, but definitely played a role in amplify and the transmission. And that, I think, also contribute to the alert of the Chinese authority and WHO. And again, same conclusion, the virus is genetically stable. And the last point is important because the first confirmed cases is December 1st, but from the phylogeny and from the evolution, they hypothesized that the first animal to human transmission may happen much earlier in November or even earlier. The second paper is on this sort of origin and continued evolution of SARS. And this paper, again, the correspond also is another formal postdoc trained in Duke-NUS and went back to China. This caused a lot of media attention. Basically, what they is propose these 2 types, the S type and the L type. So this is a single amino acid residue in that protein, which is not even necessary, the ORF8, remember in the genome I said. The ORF8, they found there's 2 amino acids in that position. It starts with a serine and change it to a leucine, S and L. And what they propose is that they actually in Wuhan start with S, but the L is much more aggressive. They use the word much more aggressive in evolution term, but the media and the journalists jump on as in virulence. Actually, the paper did not say it's more virulent. It just says it's more fit evolutionarily. And so this caused a little bit of a sort of a media kind of worrying to say, oh, the virus is mutating and we have 2 different types. And this is the latest paper from PNAS by collaboration from Cambridge and the scientists in Germany. Now, this is also sort of caused a lot of media attention is because there's so much debate, and some of them, unfortunately, is already politicized about the origin of the virus. So this PNAS paper basically says now, instead of the S type and L type, from the phylogeny, and they now have 160 genomes. And what they say, there's 3 major clusters, the A and the B and the C. But interestingly, what they said is the bat virus, the most closely related. If you use that as the ancient sort of a virus, the ancestor, sorry, not ancient, the ancestor virus, and use it as this as outgroup, then A is the most, original cluster. And the paper concluded that most A viruses actually appear in USA. So this was where, the political sort of fight, that the Chinese got excited to say, I told you the virus started in USA, which is totally not true, And this is important because 160 genome is a lot, but, compared with what we have, they'll just say 1.8 million or 1.6 million infected. This is a very small sampling size. So whatever the genome gives you the phylogeny is not a complete picture. So now I come to the work that we did at Duke-NUS. And again, Daniel Anderson, who is now in Singapore, is leading the virology, but she was my former PhD and postdoc as well. So, this is just anecdote. I'm so pleased that my former postdocs are playing such an important role internationally in fighting COVID-19. Okay, so now come back to this virus. What the point I try to make is that in Singapore, we sequenced like a 20 genome, and we did discover a major deletion event in the OIF8. Okay, so this is the OIF8, as you can see. So this is the Wuhan virus, and this is another virus in Singapore, what we call is a white type virus. And then the virus that has these 382 deletions, right? But this is the region that not only the S or L mutation happens, but in SARS, you see, they also have deletions, and some of them as large as the deletion we have detected. The point I try to make is that initially we thought this is a virus unique to Singapore, because how can, the Chinese has not reported, the US or the European has not reported. But this is really a reflection to say, if it's a rare event, and then, you only had a few hundred genome sequences out of more than 1 million, and then you are not going to detect every genetic sort of events. And this is that now we know that not only Singapore, but the same mutation has been detected in Taiwan as well. So we believe the mutation started in China, exported to Singapore and to Taiwan, maybe to other countries, but it's not a major population. That's why, you have to be lucky, with quotation mark, to detect that mutation. Okay, so now I'm going to move to this origin and early transmission events, right? I always say, just like SARS, Nipah, everything else, if you don't know the origin, if you don't know the intermediate host, it's very hard to prevent future, outbreak. So it's not a pure academic exercise, So the virus is most similar to the bat virus, so now suggests a zoonotic link. But for most of the bat viruses, really, you need an intermediate host. So I'm giving the example, I have been doing 25 years ago, we discovered and named the virus called the Hendra virus. It's a bat to host to human transmission. And a very close related virus, Nipah virus in Malaysia and Singapore, in this region in 1999, bat to pig to human transmission. SARS is bat civet human transmission. It took us 10 years to figure that out. SARS-CoV-2 now, bat, I think it was a question mark, but I think it's a small question mark. What we really don't know is that animal X, the intermediate host. There's a lot in the media and with papers. Initially say it's a snake, and I don't believe that data. And then pangolin. Now, I think my personal judgment is possible. I always think it's going to be a mammal, but I think we need more data. Interestingly, bats are hot with coronavirus. Pangolins are not until this COVID-19. But last year, there was one paper published in Viruses. Basically, they did a virome study in pangolin, and they found a Sendai virus, and they found a few coronaviruses. This, was really a paper that did not cause any attention. Even I did not read it. And then, after COVID-19 outbreak, you have many papers to say, hey, we discovered, COVID-19 related now coronavirus in pangolins. And so you have one paper, two paper, and then the third one just came out in Nature. So if you look at the phylogeny, the pangolins are red dot. It's still not as close as this bat virus. The bat is RATG13. That's the virus. And these are the COVID-19 virus. So all are based on sequencing studies. And I'm a strong believer on serology. So one criticism I have is they haven't done serology, because serology will tell you the prevalence. Serology will tell you, if you neutralization test, then how close the virus really to COVID-19. And as I said, overall, the genome identity is lower than the bat virus, but the IBD is stronger. So I already said that. And this is the menu. This is the menu in the Wuhan, Huanan seafood wholesale market. Seafood wholesale market is a misnomer because they sell lots of mammals. Of course, they sell seafood, but they sell lots of mammals. And interestingly, they sold almost everything except pangolin. I don't know whether pangolins was not sold or because pangolin was illegal, so they did not put on the menu, and then you have to, privately ask. But there was one animal which played a very important role in SARS is this civet. For those of you who could not read Chinese, I have to point out, you have to believe me. This is civet, and they sell both meat, as $70 a kilo or half kilo. And if you want the whole civet live, and that's 130 yuan. So civets are traded, and there are evidence because after they shut down the market, the scientists from China CDC went in, sampled the environmental sample. They even got a live virus isolation. So the market was heavily contaminated without any doubt. Okay, so now I'm going to go to the next topic is the law of serology, and I'm a strong believer in serology because in my career, discovery of hand gel in bats, discovery of nipple in bats, in pigs, and then SARS in civets and in bats, all led by serology before we had a sequence and a virus isolation. But of course, serology plays a role in many other aspects, diagnosis, contact tracing, epidemiology. We were the leader in the world in Singapore. Asymptomatic infection, herd immunity, seroprevalence. Now it's a hot topic. Longevity of protect immunity, right? Do all COVID-19 patients seroconvert? How long they last? Vaccine efficacy. And then I already said to search for the next reservoir intermediate host. So my lab has been really from day one. We have already developed 6 different platforms of serology because you cannot have one perfect test, always fit for purpose. And so, we haven't published any scientific paper yet, but we got a report in Science about, our really the first use of serology to do contact tracing. So for those of you who are not familiar with Singapore, initially we had a cluster, pretty major cluster, associated with a sort of a religious kind of gathering. And then we have a much bigger one later. And these two, the January 25 and the January 29 to February 9th, on the right, there's one, the case, 66 with the link. But between these two, there was a belief there was must have a link, but we could not find it. Until months later, later February, MOH, the contact tracing team, really zoomed down to a couple. And then case 91 especially is very important. But then she already fully recovered, healthy, PCR negative. But in serology and virus neutralization, she has very high titer against COVID-19. So this is a good example of filling in an important missing. Now, there's a really important question internationally about this, seemingly asymptomatic infection in children or young adults. So this is a China CDC data. Again, apologize for the Chinese on the left panel. So this, this is just population statistic. On the right, blue is a male and the pink is female. And you go from, 1 year old, on the top is 0 to 4, and you go all the way to 100 years old, okay? And then you look at the number of cases, the number of deaths, and the percentage who died. The risk factor for 80 and above is 1,000 times of, if you're 20 and younger. So the question is that, do young people get less exposure, or they're less prone to infection, or they're infected but asymptomatic or mild? So internationally now there is a serious surveillance effort, age stratified, so Singapore is a part of this consortium, so we can do large scales. All right, so this is my last slide. I'm going to finish up on the lessons that we learned in response to emerging zoonotic virus. In a quarter of century, basically, the title, I pen in a commentary in Lancet. The title is From Hangzhou to Wuhan. So that's almost my personal experience. 25 years ago, I started my career with the investigation of Hangzhou virus. And then this year, So the 4 lessons on the left, I'm not going to go each of them. But the number one is the most important because the cost postulate, right? WHO, have this IHR you're supposed to report. But what do you report? Do you report cases or do you report etiology? And for etiology, usually we want to, at least you have a virus isolation to say, that's the virus. But now we're in the era of next generation sequencing. I can tell you the doctors in Wuhan had sequence of now we know this SARS-CoV-2 on December 26th. But they could not confirm that's the etiology and they will not report. And so by the time we had the 4 sequence come out, it's January 12th. So that's 17 days of delay. So, this is for WHO, for every national leader, for scientists, we really need to think in the era of next generation sequencing, do we need to change the way we report a new outbreak? And then in China, there's a lot of this debate about, the outbreak responsible scientific publications, and then also the One Health approach, right? This is a human disease, but is it really only CDC? Like China CDC and mostly medical doctors and we should including veterinarian, zoologists, environmental scientists. And then the last one is talking about, it seems like we can never get the name right when you have an outbreak. Okay, so in addition to all the lessons we learned, and I pen in the Lancet commentary, I have 3 other lessons. One is, remember during quarantine, I was reading lots of social media, both in English and the Chinese media. It's really a double-edged sword now. I mean, even some of the scientists, I have to say, just not, responsible enough. They put out comments which I think is really misleading. But because from scientists in social media, that, goes a long way. Secondly, I'm a bat, virus person and bat biology. Every time I have to put this out, it's not bats' fault, okay? Bats have been with virus, peacefully coexist much longer than humans. So it's really the human activity, like wildlife trading, that caused the outbreak. Okay, so don't blame on bats. My last is that peacetime versus wartime. We have been predicting this going to happen, but during peacetime, you don't get enough really sort of attention. And I think that, we really need to do better. And thank you for your attention. And back to you, David.
Thank you so much, Linfa, for that excellent discussion. We're going to move now to the question and answer period, and all 3 of our panelists here are here, myself, Dale, and Linfa. So I'm going to take the prerogative of the moderator and ask the first question. You said you're a serology man, Linfa, and I'm wondering, coronaviruses are very common causes of common cold. What allows us to be certain that the serology is detecting the pathogenic coronavirus that we're worried about?
Yeah, so the COVID-19, I think, so we have, before COVID-19, we have 6 coronaviruses known to infect humans. I mentioned the SARS and the MERS, but there are 4 others that actually now is widely circulating in human. And so the good news is that those popular or the more common, almost the human coronavirus, they tend not to cross-react with COVID-19. The one really watch out is SARS. Okay, so for a country like Singapore, we have to be really careful because we have SARS survivors, and I can tell you 17 years later, they still have high levels of antibodies, and they can cross-react with COVID-19. So we have to design the test smartly to differentiate, and we have this now.
All right, thank you so much. Clarice, you have questions for us.
Yes, Dr. Allen, there certainly have been a great many questions. And as always, we apologize if we can't get to all of them. Our panel have selected a few questions they hope will benefit the most people. The first question is, why do bat zoonoses seem to be more common? For example, Nipah, Hendra, SARS, and as always, rabies. Is this an effect of human encroachment on bat habitats, or perhaps a change in bat behavior?
Good question. First, I think it's very important. As I said, bats have been, around for like 60 to 80 million years. So they're much more ancient than human. So they have already peacefully coexisted with virus. So bats basically carries much more virus than any other mammal. So that's fact number 1. And fact number 2 is that bats virus usually don't jump species until they get susceptible like a civet or pangolin or pig or, So I think that definitely that in the last, maybe a few decades, that, the farming, intensive farming, wildlife, and also climate change, all these contributes to that bats are now making more contacts with animals. So give them a chance to transmit to intermediate host. And then once it get amplified, like a Nipah in pigs, and then they get transmitted to human. So it's a 3-step. Bats carries more virus, human activity, climate change, make them now have more contact with animals. And then if they have an amplifier host, the right one, they will amplify and pass to human.
Excellent. Thank you so much. Clarice, you have another question?
Next question. In light of the close relationship that coronaviruses have with bats and humans, is there any advantage of routine bat surveillance for future renegade coronavirus?
For people like myself, yes. I need more funding to do that. Definitely. It's a short answer. It's a short answer, okay?
Oh, good. Thank you, Clarice. Go ahead.
For the third question, does the 382 anti-deletion in the 3' end of SARS-CoV-2 you reported have implications for disease virulence or spread?
that's another good question. So we have the paper now in revision, and so far we have not found any major difference. But I have to caution the audience, for the audience who are experts in the audience, that so far we have done everything in vitro in a cultural cell. So when there's no difference in the cell, does not mean there's no difference in human bodies, right? So the research is going to go ongoing. And also I'm really watch with interest to see if there's any other regions or countries where discover the same deletion like the scientists in Taiwan already found.
Clarice, please.
And do you believe that serology as measured by your assay will indicate effective immunity? When do you think we can use serology to help us know when our healthcare workers are safe to work in the front lines?
Another very, very good question. WHO, I'm going to have a meeting with WHO tomorrow night. Okay, so in order to use the serology to correlate with protection, you have to have two things you have to really know. First of all is that you are measuring the antibody, the population, what we call is neutralizing antibody. So antibodies, when you get infected, your body launch antibodies. There's two types. One is just bind the virus, but the virus can still get into your cell. The other type is neutralizing antibody. This actually prevent the virus get into cells. So this is the important. Usually, neutralizing virus is the one that most likely will be correlated with protection. So I have developed an assay. Now we're going to measure neutralizing antibody. So that's the first part of the equation is solved. The second part of the equation is much more difficult. Does neutralizing antibody correlate with in vivo protection? Okay, so this is a much sort of a more difficult question to answer, and we need a more sort of a recovered convalescent patient, and we need to monitor this for much longer than the 2 months we had right now. So we're working very hard, but to answer the second part of the question, I think we need more time and more samples.
Clarice?
It claims that SARS-CoV-2 was synthesized in a laboratory and not naturally occurring. Is there evidence to support this, or is there data that can debunk this?
Oh, I think this is a very politically sensitive topic, but there are so many papers, all the leading scientists, and actually 20 of them came out and published an open letter to science to say there is zero chance that happening.
For our last question, are new isolates of SARS-CoV-2 intermittently sequenced for mutations for purposes of determining rate of evolution, virulence changes, or association with superspreaders?
Yeah, so these are all interesting questions, but I don't think just by sequencing the genome, you can answer, all of these questions, because the super shedders or super spreaders is mostly not driven by virus. It's driven by the human immune system, okay? So that, SARS data is very clear, and also for, our Hendra and Nipah studies, the virus mutates, but there's no correlation in this kind of virus to say this virus now is more transmissible and a super shedder than the other. It's more about individuals. Some individuals just have the virus replicate to much high level and become super shedders. So it's more host rather than viral.
Thank you, Prof. Wang.
All right. Thank you so much, Prof. We really put you to work. I appreciate that.
Thank you.
We're going to move to our next section, which Dale is going to review key points and current events with us. Over to you, Dale.
Thanks, David, and thanks, Linfa. I love hearing what you've got to say and pretending that I understand so much of it. It's really interesting. I've got just a couple of things I wanted to bring up today. I'm deliberately not bringing out evidence that's designed for the previous session of this program, but this is more just to talk about things that are issues at the moment, and for so long, Singapore was called the poster child of the outbreak, and I can tell you I was always very uncomfortable in the media when this came up, and we'd say things like, things are going well so far. I think in an outbreak, it's you never want to count chickens before they're hatched, and And what we've seen is that, this virus really has the capacity to find the weakness of any country or city, and it's got the capacity to punish you. We certainly did put in a lot of work into nursing homes, the prison, the Institute of Mental Health, the hospitals, healthcare workers, things like that. I don't recall a conversation about the dormitories, and that's clearly, as I mentioned earlier, there's two outbreaks going on in Singapore now. One is in all the dormitories, and while some are more affected than others, I think that largely relates to how much testing we're doing. Sure, some might be less affected, but there's not many that are unaffected, and you're going to see the numbers really shoot up as we ramp up the testing in the dormitories. I think we're being found underprepared. You could argue the initial response has been slow, but I've been in many outbreaks, and it is complicated. In fairness, there's many players, and if you don't have a plan, you spend a lot of time even strategizing before you even get boots on the ground. So, I wouldn't be overly critical of the speed of the response, but it certainly one could argue that we could have foreseen this, but again, it's easy in retrospect. Are we the poster child? Firstly, I don't like the term. A lot of countries around the world are very proud that their curves are flattening, but actually they're shut down. And of course, curves are going to flatten when you're shut down. That's the compromise, and the clever moments will be how well you can open up without seeing a spike in cases. So, things are really going. I saw some of the questions. It's difficult to answer them all, of course, but most of the, or not most, I think about 10 of the dormitories are actually isolated off now. So they've still got the capacity to call an ambulance, but they're not. The individuals inside are not allowed to leave, and no one outside's allowed to come in. So there's a sort of a quarantine of the facilities. There's a plan that every dormitory will have a medical team in there, so that we can ramp up the swabbing and the medical care and the triage, if you like, to what sort of isolation facility. But this is going to stretch us, and as it's a numbers game. I suspect our audience is aware of the numbers, and if you look at the ages of 40 to 49-year-olds, say, or 30 to 39-year-olds, you could argue that maybe 1% of these people will end up in an ICU bed. So, the numbers are, if it's 20,000 cases, then there's going to be 200 ICU beds. So you can see how now, where we're sort of in the 1 or 2,000s, how it's just so important that we can identify these the positive COVID cases, which is not easy. It's not easy walking into a dormitory of 24,000 individuals with in 10 blocks of 2,500 each, and trying to work out where to start. So there's a huge effort going on. Meanwhile, the rest of Singapore society is having, as we said, 38 cases. It's in the last 24 hours, so it's quite disproportionate. So they're just some comments on this whole concept of poster child. I think I don't like the term poster child, but I still think we're doing well outside of the dormitories, but the dormitories are part of Singapore, so you can't deny the numbers and the impact they're having on hospitals and healthcare workers who are dealing with it. I just wanted to show you a few slides which people from Goan in the audience will love. This is an old slide from a Uganda outbreak in 2001 for Ebola. And if you just, so there's the normal volunteers here and doctors and all the people that worked on the outbreak. And I just want you to hone in on this little place here, because this is the origins of what is a famous Gohan slide about how an outbreak works, with coordination, case management, laboratory. So this has emerged, sort of evolved, I guess, over the time to what many will remember as this slide, and this became an absolute classic slide in Gohan circles of how you manage an outbreak. You know, it's not just one component. You know, the epidemiologist might think it's all about them, and the logs are thinking it's all about them, and the doctors are thinking it's all about them. But actually, it's a whole coordinated effort. So without going through that, I just wanted to point out that another fundamental of Gohan is that no single institution has all the capacity. So you really need to work together, and this shows, you know, UNICEF and IFRC and WHO and others in the field. So let me just turn those slides over to the Singapore version, and this is the dormitories, Ministry of Manpower, the dormitory operators, the SAF, Ministry of Health, and all the health workers of all the clusters and including the hospitals and the polyclinics. That the extraordinary efforts that are going on are gut-wrenching, actually. And let me put that other major slide. These are two of the blocks in two of the clusters. So each of these buildings is 2,500. So this is cluster 5. This is cluster 4. So you know, it's a small town, and this was the day before this dormitory was closed down. And each of these rooms had to be populated by the 12 people that lived there. So I put the same slide there, and I ask everyone in the audience to maybe just think about this, because this could be how you're managing. This is not one outbreak. This is millions of outbreaks in every city, every country. Or it might be an outbreak in a school that you're asked to call on, or an outbreak in a hospital, or you know, in our case, a dormitory. The components are the same, and that's the point of what I'm trying to say. There's case management, and all the, you know, we're told to send swabbing teams in, but this is just such a small part. You know, where's your manpower? What are your rosters? What are your patient workflows? Where do they go after the swab? Infection prevention and control, you know, designing the makeshift areas, ensuring healthcare worker safety. What do you do with a death? The laboratory, we're finding a lot of the people, they're not in the bed they're assigned to. A lot of people have the same name, remarkably. So coordinating all this is an effort in itself. In terms of the epidemiology, you know, it's easily forgotten when you're just doing an operation like this. But we need to track which are the infected rooms. You know, doing a spot map of 2,000 rooms of 12 people. Think about it. So tracking the individuals, managing the data, who's had a swab, who needs a second swab, whatever. And the whole policy, do you do a mass swab? These are all arguments. The logistics is huge. The medical supplies, the PPE, the drugs, pulse oximetry, cleaning, but then giving everything to the residents. They need masks. They need alcohol-based hand rub. They need cleaning equipment to keep their rooms clean because they're easily contaminated. Getting food to everybody, the clean zones for the health workers, coordinating ambulances, the security, waste management, and actually building isolation areas and makeshift hospitals, which within the car park we've done in this particular instance. But actually, I've got psychosocial support up here, which is critical. Maintenance of regular healthcare needs. There's people still have other things going wrong with them, but I still maintain that the strongest component is the community engagement and the risk communication. You know, telling healthcare workers what the plan is. Don't just say go and swab for a day and we'll tell you what you're doing tomorrow. the confined workers, getting messages to in at least 3 languages to 24,000 people is a huge task, and you can't just leave people there without telling them, what the strategy is or what their result is or what's happening. How do you get there? Do you use social networks? Do you use loud hailers or public address systems, WhatsApps, or leaflets? It's how do you get the message out there? But there is actually an existing hierarchy in these buildings. So there is someone in charge of the block, in charge of the floor, in charge of the 12-bed room. So, and then of course there's management of the media, because you don't want sort of media distractions to you don't want to not tell the media, but you also don't want it to distract from the main cause. So I probably went on a bit longer than usual, David, but I thought it fair just to let people know what's happening from my perspective in the dormitories, and also how this could be applied to other settings in other parts of the world. Thanks.
Well, thank you, Dale. That was very much appreciated to understand the complexity and the stress that this situation puts us all in. And so we very much appreciate your hard work. We very much appreciate you making time from your very, very busy schedule. It also leads me to thank Prof Wang Linfa for taking time away from his busy schedule. He's in great demand. And I also want to thank the audience for their participation. It's why we're here. So that leaves me to say thank you and to let you know that next week our speaker is Professor Kishore Mahbubani, who is Distinguished Fellow at the Asia Research Institute at National University, Singapore. He is former Singaporean diplomat and founding dean of the Lee Kuan Yew School of Public Policy at National University, Singapore. Title of his talk will be COVID-19, Has China Won? Until next week, stay safe, wash your hands, and with apologies to the Beatles, I don't want to hold your hand. Good night.